Now, in the last video, we talked about how the early Earth could be a primordial soup filled with the organic molecules necessary as the building blocks for life. So all you need now is a procedure to go from that to the life that we have today. But remember that life in its essence is basically a cell, which is a membrane surrounding some genetic material and some machinery to perform basic metabolic activities. Now, the life forms that were the original life forms were nothing like the life forms from today. They were much simpler than the life forms we see today. And we call these things protobionts, things which are almost alive. And for them to exist, what you would basically need is something similar to what cells are, a protective barrier surrounding self-replicating molecules. Now, several examples of this are seen today in laboratory. For example, coacervates are something that shows up sometimes, and there are basically a skin of water held together just by the surface tension of water surrounding a droplet of water full of proteins and carbohydrates inside. And in the human body and several other places on nature, you see several examples of simple metabolic processes happening within coacervates. For example, in digestive processes in the stomach, this happens very often. You have bubbles of chemistry basically going on. And those bubbles of chemistry are basically, if there's anything about the chemicals inside of that would be self-replicating, then you could potentially lead to life. Another example of simple structures like that is microspheres, which is basically a protein shell surrounding a bubble with water and stuff inside there as well. And there's plenty of examples of that can spontaneously generate in the laboratory as well, and several examples of that in life as well. Another example of that is also spontaneously generated solution is liposomes, which are basically lipid structures or bubbles made of lipids. Now, these bubbles, if they have something trapped inside of them, could also lead to formation of early protobionts. The key is what's trapped inside of these things. And by the way, we think that the early life form, the protobionts, were probably based of liposomes with trapped self-replicating molecules in the inside. Now, these self-replicating molecules in the beginning were probably RNA-like molecules, because the RNA that we see today is a very versatile molecule. There are many types of RNAs. Some RNAs act like enzymes. We call them ribozymes, and they speed up chemical reactions, which is crucial for the metabolic process to take place. Some RNA acts like structural components. You see that in ribosomes today. Some RNA acts like genetic material transfer. You see that in tRNA. You see that in mRNA. Some RNA doubles as carriers, tRNA. Some RNA acts like regulatory molecules. And so RNA can have multiple functions, and because of that, we think that the original molecules of life were probably RNA-based. So we have this idea that's called the RNA world hypothesis, that the first original life forms were basically protobionts, which had one of these structures, one of these protective bubbles, most likely a liposome, and inside of it you had these self-replicating molecules which were probably RNA-based. Because you see, from a single-stranded RNA molecule, it's easy to get to DNA. All you need to do is to duplicate the RNA and then hold it together in a double-stranded molecule, and now you have a simple DNA molecule. Now, if that simple DNA attaches to another simple DNA molecule, you start becoming larger and larger, and you start getting to the idea of a large molecule composed of smaller chunks, or called genes. And then to get from that to proteins, well, ribozymes, which RNA, can help us to synthesize proteins. And so you can have all of that be possible. And that's what they're trying to describe in the bottom there, that ribozymes can catalyze chains of molecules such as proteins from environmental pieces. So all of which life is could have started from such a simple thing which we call a protobiont, which is basically a protective bubble surrounding self-replicating molecules. To understand a little bit better about how that process could possibly happen, watch the other videos that I have on the actual history of life. In the beginning of that, we talked about the origin of the first protobionts. So that's how life actually starts. And I hope you understand a lot. And in the next videos, we're going to be talking about some evolutionary topics that talk about how we go from that to the life that we have today. I'll see you guys then.